my title, A Primer on the Human Brain and the Possibilities of Drug Interactions. I thought that was really long, and so like I figured, well, what does it all mean? What is the point that I'm trying to get across? So I made a more colloquial title. Here we go, the brain. It's fucking complicated, and why that can be very fun to the average student. So you know, like there's so much information out there, but I think with a little bit of depth, that you can get some really stimulating conversation out. And so basically I was prompted to make this whole presentation by the fact that I love to relate my stories to sort of biological principles. And like other people love to relate their experiences in biological principles. But like we don't always have the vocabulary or the know-how to sort of explain those things. And so hopefully this will give you sort of an entranceway into that. Okay. So the purpose is to explain some of the basics of neuroanatomy, which is basically the structure and function of the brain, and to give all of you guys an entrance point into the discourse of neuroscience, so that when you go out and you are researching your favorite drugs or you're researching something interesting about drugs, you can sort of read that paper and say, oh, I kind of recognize some of those words. It has to do with this, it has to do with this. And so that's what I want to do. So relationship to psychoactive drug use. So we're talking about the central nervous system here. Neuroscience is a huge field. It's extremely vast and complex. You may hear terms like neuroanatomy, electrophysiology, molecular biology, behavioral psychology, and cognitive neuroscience. And you know, there's a big movement now to sort of merge those fields of the psychology with the biology. And what exactly is the mind in biological terms? You know, it's being defined a lot in psychological terms. Okay, so topics. Neurons and glia, which are we're going to start small, we're going to go big. And so we'll start with neurons and glia, which are sort of like the cell types that we're seeing in the brain. We'll talk about synapses, how they all join together. The vasculature of the brain, which is all those fluids that are sloshing around your noggin. The structural organization, so those are just like regions and how they all get connected. And then I want to end with a simplified model of things because I'm going to get confused halfway through. You know, I'm going to start talking about things in the structural organization and it's confusing, and so I just want to say like a sort of last words how the brain works. But then at the same time, go in there and say there's tons of questions you can ask on top of that. Because maybe most of you are scientists, I'm a scientist, and certainly that's the most interesting aspect to me. The fact that there's an infinite number of questions you can just start directing at this material and find that it has holes everywhere. And your experiences may correlate to something, but who knows what's actually happening. Okay. Oh, but before I begin, I want to have a disclaimer and sort of talk about my sources. Because like I'm talking about this like a really broad sort of thing, I don't want anybody to get confused. I'm not a neuroscientist, I'm actually in microbiology. And so all of this information I just gathered from textbooks and such. I didn't look up everything, I just looked up the textbooks that had the main words in the title. And anyway, so I used that. And I didn't double check a lot of the accuracy. And so this might be really critical because most of the sources I use are pretty up to date, but the field is rapidly changing. And so, like, don't quote me on too many things because there might have been discoveries later on that would, you know, sort of invalidate some of the things said now. Like, for example, there's this theory that different parts of the brain have different functions, but like even that's sort of coming under fire with new research. All right. So let's start with the neurons and the glia. So basically this is a wonderful picture of rat thalamus, just a part of the rat brain sort of in the middle. And there's two major cell types here, glia and neurons. And so the neurons are the ones that we all think about when we think about brains. They're stained green, probably a green fluorescent protein. And then the glia are stained red, probably red fluorescent protein. And then the blue, those represent the nucleus. It's a stain called DAPI. So those are the two main components, and I guess first what I'll talk about is the glia. Glial cells outnumber neurons 10 to 1. So when you think about your brain, it's full of all these neural connections. Actually, there are tons of other cells there that aren't even neurons at all. And they support neurons in a variety of fashions with a variety of functions. So we have the microglia, which mediate cell tissue repair and can phagocytose debris and pathogens. Basically, they're the resident immune system. So I don't know if you guys know, but the central nervous system is cut off from the rest of your body because potentially dangerous inflammation will just blow up your brain. And so they don't really have their like immune system running around there. They have their own homebrew immune system, the microglia. And the macroglia include astrocytes, which have a structural function. Basically, they're thought to be like one of the first cell types to emerge in your brain. So first your brain is this big ball of astrocytes, and then the neurons start projecting through. And so anyways, there's a lot of research here in development because certain molecular signals will be signaling to the neurons where to project. You need to be here, you need to be over here, and so they're very important in that sense. They also metabolize a lot of things. They provide some nutrients to the neurons, and they also play a role in the vasculature, as we'll see later. Oligodendrocytes and Schwann cells, which form a myelin sheath around the axon. So I didn't even say anything about axons, but very quickly, axons are these like big long projections that are coming out of the neurons. They send signals, and myelin is sort of this like fatty white substance that coats the axons, making those signals go faster. So like you'll have actually it's really cool because myelination actually occurs like around our ages. Like it's not totally done yet. So like brain development is still going on right now. And you're myelinating the connections that you're using most often, and then you can use those connections as fast as you want. So I think that's pretty cool. Myelination. And endodymal cells, they mediate the flow of chemicals from the cerebral spinal fluid. So as I said, the blood is largely separate from the central nervous system, and so these are very important in sort of gating. Like you still need fluid in there. It's not like totally separate. It doesn't eat its own food and anything. So you need a system to sort of mediate the compounds from one liquid into the next. And that's how we do it. And a wonderful picture, I think. Oh, yes. The neuron, it's in yellow, and then we have the Schwann cells or oligodendrocytes in the blue. You can see that they're attaching to those axons, putting myelin sheaths around them, and also astrocytes just in the background. They do a lot of other things. Okay. Next. Neurons. So neurons, the basic cell types. This is what we're going to talk about in terms of all those that information sending. These are the cells that are sending information all around your body. So they receive information from the dendrites. They process in the soma and then signals by the axon. So three main parts. The dendrites are like these kind of like fuzzy little things. They're often extremely branched, and they branch off of the cell body, which we'll call the soma. Some people call them the nuclei. I'll probably say all of those words in switching, whatever that means. So axons go out, and they can be incredibly long. Axons in your brain can reach all the way down to your toes. At the same time, they don't have to be long. They can also be very branched and sort of all over the place, as you can see with like certain axons of the thalamus. And so they're classified morphologically, so that's by their shape. And so here we have three shapes: multipolar, bipolar, and unipolar, but also by molecular identity. And so this is something that means a lot to me because I am sort of in the molecular field. And molecular identity means that there's certain types of molecules, receptors and stuff that are sticking out. There's also certain types of enzymes and things that are expressed within these cells. And the brain is one of the most heterogeneous organs in terms of cell types. There are so many different cell types. Like the cells of your cortex, I'm going to explain later, 6 layers. Every layer has a different cell type. Every localized layer has like different cell types. Like this spot over here is something different than this spot over here. And like you all guys know the Canadian heritage moment, right? You like poke their brain, she smells burnt toast. I mean, that's so specific. And part of the reason that arises is because of their molecular specificities. Okay. So they're extremely morphologically variable. You can see some look like tufts of whatever, some branch off like this, and it all depends on what they're doing, where they're connecting, and what their purpose are. And I'm not going to go into any of that because, frankly, it's just too deep. But I will go into some main sort of ideas of how they form the circuits. So information from receptors is carried by bipolar and unipolar neurons to the central nervous system. So there's the central and there's also the peripheral. And so in the peripheral, we have like baroreceptors in our hands to feel pressure. We also have photoreceptors in our eyes. The list goes on and on and on. But what's sort of important is they receive some sort of physical stimulus. They turn that into sort of a chemical stimulus that then goes to a neuron, goes to your spinal cord, goes up your spinal cord, some processing in the brain. You think about it, output comes out. So that's that. Multiple neurons carry information to multipolar neurons, which process the signal and pass it on to more multipolar neurons. So this is pretty crazy because that means there's a lot of like processing that's going on in the brain. It means that it's not just as simple as saying, you know, this touch goes to this cell, then this cell goes to this area, this cell goes to this area, and then goes back. It actually means that it goes into one cell, multiple stimuli can go into one cell. That one cell can then maybe touch its partner, which touches itself again, that like branches off all over the place. Basically, the networks can get very complicated. And then multipolar neurons in the central nervous system can project all the way to the muscles to initiate voluntary action. So as I was saying, they can be very long. The axon projections can be extremely long. And so you may have heard gray matter versus white matter. Gray matter is basically the nuclei, and white matter is the tracts of axons that are moving through your body. They're sending the signals to the gray matter, which processes them and sends them out again. Okay, synapses. So how does that information really jump from cell to cell? And it all comes down to these synapses. They're the mode of neuron to neuron communication. So as you can see, you've probably all seen a picture like this before. When the appropriate action potential stimulus causes signal flow, the appropriate action potential stimulus causes signal flow across the synaptic cleft. So basically, when the cells, so this is an axon, and this is a dendrite. And so when something exciting happens to your cells, there's this whole event, it involves ions and charges, but what you need to know is an action potential moves down the axon. And when it hits this presynaptic membrane, it's going to release a whole bunch of neurotransmitters based on the identity of the cell. And those neurotransmitters will cross this little cleft, which is, by the way, not a gap at all. There are proteins that do span the whole thing, holding it all together. And they're going to hit the postsynaptic the membrane, and that will either increase or decrease the probability that the neuron that it's attaching to is going to fire. So because of the increase and the decrease, you can call some neurons excitatory and some neurons inhibitory. So, you know, some neurons are going to say, go, go, shoot, shoot, fire off, like information. Some neurons will say, wait, too much information, slow down, like, don't get too excited. And so, you know, we can think about that like seizures, right? Like people who have seizures, they're just like, I think they lose the inhibitory sort of aspect, and then they just like seize up. But anyways, there's a lot of reasons for seizures, and that might just be one of them. Anyways, the signals in the form of neurotransmitters. I think people who like to discuss drug use love to talk in neurotransmitters because like they are responsible for a lot of gross changes in our personalities or our behaviors. So really quickly, you probably heard dopamine is reward, acetylcholine is association, serotonin is mood, noradrenaline is stress. But there are a whole whack of other ones. And also, those aren't the only functions of those neurotransmitters. Like acetylcholine, if you know, also contracts your muscles. But only in the brain is it really associated with association. That sounds like a ridiculous statement. Okay, and finally, very complex action potential firing. So I took that from a paper or something. But basically, when you say action potential firing, sometimes you think switches on and off. But it's more than that. There's frequency and amplitude information encoded within action potentials. And so you could have like fast, big firing neurons, or like slow, short firing neurons, or like slow, big firing neurons, fast, anyways, all those combinations. And that sort of information is also being synthesized by the cell body in order to figure out if it's going to fire. Vasculature. So two fluids circulate the brain in separate systems. One is blood. We all have blood, we know what it does, oxygen and glucose. But basically, it's the basis of fMRI, which is this technique that says, oh, the brain is working because it's getting, there's blood flow in this area. Therefore, that area of the brain is working. And so they'll test it in different scenarios. They'll be like, oh, when the guy throws the ball, these areas of the brain are working. So those areas of the brain are associated with ball throwing behavior. Well, of course, they're going to say something more precise than that. This is a picture of the arteries or something that are going into the brain, arterial blood flow. Basically what you need to know is blood flow is constant into your brain region, and it's only the local areas that the variation occurs. So it's like local dilation of tiny capillaries in different areas. And how that happens is, as I mentioned or hinted before, is via the astrocytes. So like astrocytes are these cells that sit by the synapses, and maybe they see a lot of activity going. They're going to pick up those chemical signals and then start the dilation or the constriction of the capillaries in that area because they need blood, they need oxygen. And the other one is the cerebral spinal fluid, which is basically this like, it looks like lymph. Maybe no one's even seen lymph, but it looks like clearish stuff. And it's basically blood plasma because the ependymal cells, they filter out